Look at this automated, sterile filling line. Thousands of vials are processed here every hour, moving at a speed where a single microscopic error can replicate across an entire batch before an operator even notices. In most manufacturing sectors, a 99% success rate indicates highly efficient operations. In pharmaceutical production, a 1% failure rate is a public health catastrophe. Absolute zero-defect compliance is the only acceptable baseline. Historically, some facilities relied on external regulators, like the FDA and EMA, to act as their primary quality check. The FDA is the United States Food and Drug Administration, and the EMA is the European Medicines Agency. Both are external governmental bodies governing drug safety and efficacy. Relying on these external agencies to catch operational mistakes is a reactive strategy. If a government inspector finds a manufacturing deviation before the internal team does, the facility's quality control system has already failed. To prevent this, facilities must engineer their own proactive, internal diagnostic mechanisms to catch and correct deviations long before an external regulator ever steps onto the manufacturing floor. The pharmaceutical industry formally defines this internal diagnostic mechanism as self-inspection. Self-inspection operates entirely within the facility's quality management system, or QMS. A QMS is the formalized framework of policies, processes, and procedures required to plan and execute safe drug manufacturing. Self-inspection functions as a dynamically engineered feedback loop designed to detect, isolate, and remediate systemic manufacturing deviations before they escalate into public health crises. Evaluating GMP compliance across a massive, modern pharmaceutical footprint is a complex logistical problem. GMP stands for Good Manufacturing Practice, a regulatory system ensuring products are consistently produced and controlled according to strict quality standards. To structure this evaluation, facilities use the WHO TRS 908 guideline as their foundational baseline. WHO TRS 908 is a technical report series published by the World Health Organization that establishes specific guidelines for internal pharmaceutical quality audits. This framework mandates the rigorous evaluation of all production and quality control vectors, requiring internal auditors to examine every operational layer without exception. The first two critical areas stress-tested under this framework are personnel and premises. Auditors verify that employees possess up-to-date training matrices, and they confirm the physical facility is clean, maintained, and operating precisely within its environmental parameters. The next two areas focus on documentation and sanitation. This involves scrutinizing production records for strict data integrity and challenging the facility's contamination controls to ensure sterile conditions are actively maintained. By simultaneously evaluating personnel, premises, documentation, and sanitation, the inspection targets the core intersecting variables of drug manufacturing. Rigorously evaluating these baseline parameters provides the objective data needed to mathematically prove operational readiness, rather than waiting for an external regulatory trigger to uncover a critical flaw. This requirement raises an immediate logistical question: who within the facility is structurally qualified to execute such a demanding internal stress test? We find the answer in a universal consensus shared across major regulatory guidelines, including EU GMP Chapter 9, US FDA 21 CFR 211.180, and PICSIS. These global regulators explicitly mandate a design constraint known as structural independence. The rationale is straightforward. Operational personnel are inherently incapable of objectively auditing the very systems they manage and are evaluated on day to day. Look at this organizational chart. If a manager audits their own department, the structure flashes red, indicating systemic failure where biases hide. By rerouting auditing authority to an independent quality assurance unit, the chart highlights green, eliminating systemic bias from the reporting equation. Without the strict design constraint of structural independence, internal audits are merely self-validating loops that actively mask underlying systemic risks from senior management. Now, with the organizational reporting structure secured, we can examine the actual floor-level execution mechanics of the self-inspection. This outlines the self-inspection process as a continuous loop: plan, audit, report, and follow-up. First, audit schedules are risk-adjusted. Second, the audit requires floor-level data extraction. Third, the report categorizes every observation strictly as a critical, major, or minor defect. This strict risk categorization serves a specific engineering purpose. It prioritizes the allocation of time and capital resources for the final, most crucial step of the cycle. Effectively running this rigorous four-step cycle translates qualitative observations from the factory floor into actionable, risk-weighted engineering data. Many facilities identify deviations and document them in exhaustive detail, yet fail to implement the engineering changes required to prevent recurrence. This gap between identification and remediation represents a frequent and severe failure point in pharmaceutical manufacturing. The audit report serves as a collection of raw data, outlining the symptoms of a problem, the first step in a larger corrective process. To cure the problem, facilities rely on the corrective and preventative action, or CAPA, subsystem. Zooming into the follow-up node reveals three mandatory phases: root cause analysis, action implementation, and effectiveness check. This closed loop is the only legally recognized operational deliverable. Failing to complete this final step carries severe legal and technical consequences during an external regulatory inspection. If a facility cannot provide documented proof of remediation through a rigorous effectiveness check, the preceding internal audit is technically and legally void. An audit report identifies where the system is hemorrhaging, but only rigorous CAPA integration actually stops the bleeding. This makes the follow-up phase the definitive metric of a QMS's health. We have mapped out the mechanical design of the QMS loop, but machinery cannot operate in a vacuum. It requires specific environmental workplace conditions to run. This environmental condition is known as the culture of quality, and it forms the foundational layer upon which the entire QMS architecture rests. The self-inspection loop has an inherent systemic vulnerability. It is entirely reliant on accurate, transparent human reporting to function. If a facility enforces a punitive reporting culture, operators are naturally incentivized to hide deviations out of fear of reprisal or job loss. Hiding those deviations actively starves the self-inspection loop of the accurate raw data it requires to function, triggering a complete systemic collapse. The solution to this vulnerability is structural. An effective self-inspection protocol must rigorously target the faulty process, not the individual person running it. The investigation must prioritize uncovering the systemic why behind a manufacturing error, rather than launching a hunt for the individual who. A facility integrating mechanical independence, strict CAPA rigor, and a non-punitive reporting culture demonstrates genuine quality ownership. For the regulatory and quality assurance professionals watching, how do you integrate complex CAPA tracking with your internal audit schedules? Detail your specific strategies in the comments below.